Isn't there just something satisfying about that diagram, though? I mean, looking at that, thinking, that's awesome.
It's complete. Hold on. I'm getting this right too.
Right?
I'm gonna get in everyone's picture and do a panoramic real quick.
Do you need more?
Yeah. I'm gonna do that.
I think you should.
Okay, I got 3 pictures, autographs.
Okay, I'm coming up now.
So before this, does Photo Systems 2 and 1 happen, or is this? Sorry, I'm kind of confused about that.
Good question. So what's the relationship then between cellular respiration and photosynthesis? Really is what Adam's question is. So it's not a dumb question at all. The first question is, do plants go through cellular respiration? And the answer is absolutely they do, right? Because they need to grow as well. And the way that they're growing is by creating some cellular energy, ATP, and then using that cellular energy for growth and damage repair and things like that. So where do they get their sugars from then to go through cellular respiration? Photosynthesis. Yeah, exactly right. So they go through photosynthesis, they create a whole bunch of sugar. In fact, they create more sugar than they actually need in the moment. And then they store that sugar in a variety of ways. They can store it in their roots. That's what tubers are. That's what potatoes are. They're just a bunch of glucose all connected together in the form of starch. They can store it in fruits, for example. And then they're able to use those stored sugars when they need them. Now, remember how silly I thought that was? Because really the product of photosystem 2 and its associated electron transport chain is what? What are we making from that second electron transport chain?
Proton gradient.
We make a proton gradient. That proton gradient is used to create what?
ATP.
ATP. And so with photosystem 2, its associated electron transport chain and ATP synthase, they can make a whole bunch of ATP. So the question is, why do they need to go through all the dark reactions at all, right? I mean, if they're able to just photosynthesize and make some ATP, why don't they just do that instead of that long, drawn-out citric acid cycle? Or, excuse me, the Calvin cycle. And the answer is, ATP is incredibly unstable. You can't make ATP and store it. It'll fall apart. So they have to create some energy that's in a storable format, and that storable format is sugar. Yeah, Adam.
So is the Calvin cycle, remember you have 3 carbon, where we start off? Is
So
it
3 carbon dioxides, yep, and you're fixing them,
Yeah, fixation.
and then you're reducing them, and then you're
And that
reclaiming them.
happens after photosystem 2 and 1.
It has to, right? Because to drive that reduction reaction, we need to have the ATP and the NADPH.
That makes glucose, which then makes this.
Right, yeah. So ultimately, those G3Ps that we're making from the dark reactions make a glucose, which they can then spin through cellular respiration. Other questions about this? Okay. So I won't erase this just yet. I'm just going to start into our PowerPoint presentation.
Chapter
7. So we're going to finish up chapter 7 really quickly. What we need, oh yeah, Linda.
Can you go over that last part one more time of the chemioosmosis of when the ATP gets transferred? How do you get the 34 versus the 36? Can you go over that one more
Yeah,
time?
you bet.
Sorry.
No, that's not a problem at all. So we know that all of this ATP being made over here is a product of redox reactions along the electron transport chain, right? It's creating a proton gradient that ultimately is spinning ATP synthase and making some ATP. So the problem is not all of our electron carriers are inside the mitochondrion, right? There's one that's in the intermembrane space, or 2, that are created in this step. We need to get the electrons from here into here. And the way that we do that is by passing that electron via a carrier protein
Okay,
inside.
okay.
Now, when it passes inside, that electron can either be passed to an NAD plus or to an FAD plus.
Okay,
If it goes to an NAD plus, we know that we're going to maximize our ATP generation or synthesis because that electron gets dropped off at the top of the electron transport chain. If, however, it's passed to an FAD plus, then we're going to get fewer ATP from that because that electron is dropped off lower on the electron transport
Okay.
chain.
that makes sense.
Okay, good.
Thank you.
You bet. So, let's talk for just a few minutes about fermentation reactions. We're going to talk about two different fermentation reactions. The one that we use in the culinary arts and one that we actually use in our bodies. So fermentation is really anaerobic respiration. And what's happening is we're breaking down a little bit of sugar to make a little bit of energy. And where does anaerobic respiration actually occur? In which stage of cellular respiration is anaerobic? Yeah, it's just this one right here, right? So with fermentation, what happens is we make some pyruvate, but we need to get our electron carriers back so that we can continue to break down glucose. And so with fermentation reaction, NADH, instead of ultimately reducing the electron transport chain, we use it to reduce pyruvate or something similar. There's a finite amount of NAD plus in the cytosol. And so to continue to go through anaerobic respiration, we need to reclaim that. So we need to drop its electrons off on something other than the electron transport chain. So in ethanol fermentation, what happens is this reaction right here. We make our pyruvate molecule, or rather some bacteria make the pyruvate molecule, and then they break one of those carbons off, kind of like what happens in the intermembrane space in that first step of the Krebs cycle. A carbon molecule gets broken off, and that remaining 2 carbon molecule gets reduced by NADH, and that becomes acetaldehyde, which is then manipulated into ethanol. So if you think about like sparkling wines, or if you think about beer and sparkling wines are carbonated because when those yeasts are fermenting the sugar, they're breaking off a carbon and creating carbon dioxide. That bubble, that fizz, the head that you get on a beer, is a product of that fermentation reaction, carbon dioxide being produced by those yeasts. And then the alcohol in those drinks is created by this process, taking acetaldehyde and reducing it into ethanol.
So that's why like alcohol is like bad for you.
Well,
You're
alcohol's,
like putting too much like carbon and all that stuff. I don't know.
alcohol's, well, drinking carbon dioxide and fizzy sorts of drinks, all fizzy sorts of drinks can be kind of bad for you. It can cause kidney stones, they suggest. But the other reason that alcohols are bad for you is because ethanol is a poison, and you're actually doing cellular damage. That's why it makes you feel good, because you're killing yourself. Yeah.
I remember that.
Okay, so that's ethanol fermentation. Now please remember what happens is one of the carbons is broken off with pyruvate. That remaining 2 carbon molecule, which is acetaldehyde, is reduced by NADH. It picks up those electrons and it becomes ethanol. Okay? Now, lactic acid fermentation is what you and I actually do. Has anybody ever like gone out and run a really long distance and the next day have your muscles just ache, be so sore? That's a product of this lactic acid fermentation, or so we used to think. With lactic acid fermentation, what happens is we go through glycolysis. We get to this point, right, where we're so aerobic that we can no longer keep up with the oxygen demand of cellular respiration. We can't create enough ATP. And so part of our cellular respiration becomes strictly anaerobic. And anaerobic produces something called lactic acid. And lactic acid is produced by pyruvate itself being reduced by NADH. So NADH comes along and it drops off its electrons onto pyruvate. And then that pyruvate becomes lactic acid. and it truly is an acid. The question is, why does that happen? And again, that happens because we need to reclaim that NAD+ so that we can continue to break down glucose just a little bit to make our 2 ATPs for every glucose molecule. Now, what we used to think, right, was that those sore muscles after doing something like really aerobic was a product of lactic acid buildup in our muscles. In actuality, that's not what's happening. Really, that soreness is a product of a buildup of potassium. Lactic acid is metabolized really, really quickly by our bodies. And so that soreness really isn't a product of lactic acid damage. It's really a product of potassium ions building up in our muscles, which is why, once you get up, sorry, just a second, Linda, which is why when you get up the next day and you're really stiff and you start moving around and flexing your muscles a little bit, that stiffness and that soreness goes away, right? Because you're using those potassium ions again for muscle contractions and for neural communications. Linda?
Is that why they say eat a banana before you go work out?
Yeah, potassium is supposed to prevent cramping.
Okay.
Yeah. So with cramping, what happens, right, is your muscles seize up, which means that they're being overstimulated, and that can be a product of miscommunications between neurons.
Oh,
So you're trying to counteract that idea. Okay. Questions about fermentation reactions? All right, so the last thing I want to talk to you really quickly about is how it is we handle other molecules inside cellular respiration rather than just glucose. Now, we use glucose as an example of cellular respiration because it goes through all of the stages of cellular respiration. But the other 3 molecules don't. Now, we're not going to talk very much about nucleic acids because if you're using nucleic acids for cellular energy, if you're burning up your DNA to create some energy, you're in the world of hurt. There are other problems going on. But you commonly use proteins, unfortunately, and fats. So with proteins, the way that we're going to get this into the cellular respiration cycle or process or reaction is we're going to take individual amino acids. Remember, a protein is a polymer of amino acids all stuck together by dehydration synthesis, right? So we're going to take a single amino acid. We're going to hydrolyze that amino acid, and then we're going to remove the nitrogenous compound, the amine group. And then we're going to take that remaining 2 or however many carbon molecule and put it right into the Krebs cycle. So we're going to skip glycolysis altogether. Maybe I should draw an amino acid just so you guys can remember what they look like. So amino acids had a central carbon, right? And they were attached to a hydrogen and then some side group on the top. And that side group is what gave them their individual identity. Then on one side, they were attached to a carbon, like that. And then on the other side, they were attached to a nitrogen, like that. So what we're saying is, we're going to take a long protein, and we're going to cut just this part off. And we're going to handle amino acids individually. But before we can put this amino acid into cellular respiration, we're going to take off our nitrogen compound, and just this remaining 2-carbon chain is going to go in to cellular respiration. And it goes in at the Krebs cycle stage, right? Because this is what happened. We created a 2-carbon chain right before we went inside the inner membrane of the mitochondria and attaches it to oxaloacetate. So this 2-carbon thing gets attached to oxaloacetate, and we're able to create some ATP from that. More interesting is the oxidation of fats. And what happens is a process called beta oxidation. And you'll remember that fats looked something like this. There was a glycerol molecule, there was a 3-carbon chain, right? And then this 3-carbon chain is attached to 3 fatty acid chains. And those 3 fatty acid chains were just whole, like, huge chains of carbons attached to a bunch of hydrogens. And you can have like 16 to who knows how many carbons all attached together in those fatty acid chains. So the first stage in the oxidation or the metabolism of fats is we remove the fatty acids from the glycerol molecule. And then we do something called beta oxidation. And beta oxidation is taking 2 carbon segments and chopping them off, and then it's taking them right into this stage. So you attach them to acetyl-CoA and then run them through the Krebs cycle. So think about the relative number of carbons on a fat, a triglyceride, and a sugar. There are lots more carbons associated with a fat, and there are much larger molecules too. It's estimated that there is twice as much energy stored in a gram of fat as there is in a gram of sugar. Lots more potential energy. Okay, I know I kind of blew through that quickly. Do y'all have any questions about metabolizing either proteins or fats?
Well, we see a lot of this on the exam.
A lot of this stuff?
Yeah.
If I ask you about this stuff, I'll probably ask you about like beta oxidation, what that actually means. Where these molecules are entering cellular respiration. Both of them are entering really at the Krebs cycle, yeah.
Can you explain one more time the beta oxidation?
Sure, so with beta oxidation, and I'll draw a fatty acid chain up here, what we get, right, is a carbon that's double bonded to an oxygen. And you're not going to be able to see that because you're kind of behind the projector. Does anybody still need this diagram? Because I need some board space. Okay, so fatty acid chains look something like this. We have our carbon that was attached to a carbon double bonded to an oxygen, and then a whole bunch of carbons, and this is glycerol right here, attached to a whole bunch of carbons like this, right? And each of those carbons was bonded to two hydrogens. So with the metabolism of fats, what we're saying is we remove the glycerol molecule, and then we start the beta oxidation stage, which is taking a 2-carbon chain and putting that into the Krebs cycle, and then taking the next 2-carbon chain and putting that into the Krebs cycle. That's the beta oxidation.